All right, in this video we're going to talk about uric acid and its role in human health and disease. But before we go into that, we need to have a little background on uric acid. All right, I often use the words uric acid and urate interchangeably, but they are technically not the same thing. Okay, it turns out uric acid is the conjugate acid of urate. Okay, uric acid can actually give up a proton and become urate. So if uric acid is deprotonated, it becomes urate. Okay, and they both exist to some extent in solution. And you say, okay, well, I understand how it ionizes in solution, but it turns out that uric acid has a lot of important properties that most people don't know about, and it's kind of cool, to be honest. All right, we have a concept in biochemistry and biology and nutrition, etc., free radicals, and we know they're bad for us. We don't want a lot of free radicals. Here's a free radical designated R dot. That's our radical here. We don't want that radical. It can cause damage to proteins, DNA, membranes, etc. We don't want them. We want to get rid of them. We want to neutralize the radical. And depending on what cellular compartment we're in, we have different antioxidants. An antioxidant is a molecule that neutralizes the free radical, gets rid of it basically. All right? And typically antioxidants that you've probably heard of are vitamin E, vitamin C, which is ascorbate here. But one that you may not have heard of is uric acid. It turns out uric acid is the most important antioxidant in the blood. You actually have a lot of uric acid in your blood, and its function is to be an antioxidant and protect against free radicals. Not something most people know, and it's actually very cool and very important. Why do we have so much of it in the blood? Well, you can have different theories, but the fact is, remember from the previous video I mentioned that the enzyme that would normally degrade uric acid in most organisms, urate oxidase, we don't have. We don't have urate oxidase. In humans and pretty much every upper level primate species, the gene for urate oxidase is held transcriptionally inactive, meaning we don't make the protein or enzyme. We don't make urate oxidase. We have the gene for it. We could make it, but it's not active. So we don't get any urate oxidase, and so we just have a ton of uric acid floating around. And the body has two things it can do with it. It either excretes it normally, or it'll keep it in the blood. And it's a combination of both of those, right? It's going to excrete as much uric acid as it needs to, and then it's going to keep a certain amount of the blood to scavenge radicals. And you see here that our radical, is going to donate its electron, or actually, it's actually going to, pick up an electron from uric acid, which is going to generate this radical on uric acid, or urate. And you might say, well, now we just moved the radical. Why does that matter? That doesn't help us. We just moved the location of the radical, and it does help us. The reason it helps us is the radical electron is now tied up in resonance. When you tie a radical electron up in resonance, it's rendered basically neutralized. Okay, it can't really hurt anybody now. Okay, and that's what all free radicals do. They get tied up into resonance by antioxidants. If a radical got tied up by ascorbate, as it is here, this radical is tied up in resonance. Vitamin E does a similar thing. So antioxidants, the way they work is they tie up radicals in resonance, which renders the radical useless. Or we don't want it to have use. We want it to be useless. We want it to be neutralized, all right? And in the blood, it turns out both urate and vitamin C or ascorbate play a huge role in protecting against free radicals. All right, so that's rather interesting. Now, in terms of normal elimination, so when we want to get rid of the urate, we just get rid of it through our pee, our urine. I want to show you one thing about this that I find rather interesting. Notice we can move uric acid into the tubular cells, which is going to be to save it. In other words, so we can use it, but we can also put the uric acid back into the proximal tubule, which is then going to be in the filtrate, which is going to be in your pee, basically, okay, your urine. Now, the fact that you can move uric acid back into the tubular cells and ultimately into the interstitial space to be used again, and the fact combined with the fact that you can move it back into the tubule, indicates that for humans, it's very important to maintain uric acid levels. The fact that I can both excrete it, but yet pull it back into the cells to reuse it, maybe in the blood, really indicates, to me at least, that regulating its concentration in the body is extremely important. That also has a lot of implications in health, because most health problems are a disease of dysregulation. Okay? What happens, for instance, if maybe I can't excrete, in other words, or secrete, I should say, in terms of kidney terms, what happens if I can't secrete the uric acid into the tubule? Maybe I can't secrete it well enough, so it starts building up and building up in the blood. I can't excrete it as well. That's going to give you something called gout. Now, in the liver, in your hepatocytes, you have peroxisomes, okay? In most organisms, peroxisomes, if you actually look at them, they have a crystalline core. The crystalline core of most peroxisomes it basically is an enormous amount of urate oxidase, okay? This is where these enzymes are kept, and the urate oxidase then is going to catabolize that urate, okay? Now, we don't have urate oxidase, so if you actually look at a human liver cell, and look at its peroxisomes, it doesn't have a crystalline core because we don't have urate oxidase. That's what the crystalline core is. It's urate oxidase, okay? So we can't get rid of the urate, and if we have a problem with eliminating it, secreting it into the filtrate to urinate it out, we have a problem. We're going to start building it up and up and up, all right? And that causes dysregulation because we can't catabolize it. So we get something called gout. Now, molecularly, what it is it's crystallized uric acid. So a certain number of uric acid molecules start hydrogen bonding to one another, and that basically renders the uric acid insoluble. Normally, uric acid, when it's just by itself, it's actually relatively polar, okay? It's polar, and that's why it's in the blood. It can float freely in the blood and perform antioxidant functions. However, when it sort of polymerizes like this, these green ones are the uric acid, it's supposed to be what they're representing, they start hydrogen bonding to one another, and when you have a large amount of these hydrogen bonds, it renders the entire crystal of the uric acid insoluble, okay? It makes it insoluble, water insoluble, that is. And when these uric acid crystals, they start crystallizing and get bigger and bigger. When they start crystallizing, they become more and more insoluble. And being insoluble, they tend to sink to the lowest possible places they reach, which happens to usually be in the knees and the toes. Now, I can't tell if this is a finger or a toe. Whatever it is, this is what gout actually looks like. Sometimes they actually have to physically cut that part of the body open and remove the crystals. This is gout. Gout is extremely painful. It's a horrible disease, painful, and there really isn't a good treatment for it. You actually have to surgically get the gout removed. And what they're actually removing are these uric acid crystals. And the reason they crystallize like that is because when they start hydrogen bonding to each other, when there's too much uric acid, the fact that they're crystallizing and hydrogen bonding to one another makes them insoluble. They're no longer soluble in the blood, and so they do this, okay? Now, we're not going to go too much into this right here, but just to give you a little bit of implications maybe in the future of health, these irritant particles right here, those are crystals of gout. And what is known actually is that these gout crystals can actually be intaken by certain immune cells, okay, phagocytes. And it turns out that these crystals actually signal the production of reactive oxidative species. They signal caspase activation. Caspases are enzymes that promote cell death. They kill cells, and you also get inflammasome activation. So it is leading researchers to believe that gout actually promotes reactive oxidative species, cell death, unwanted cell death, that is, and inflammation. Okay, so just an interesting place to take in the future. All right, so hopefully that gave you a little bit of intuition on gout and uric acid implications in human health and disease. In the next videos, we're going to go over pyrimidine catabolism, and we're going to start with thymine. Thank you for watching this video. I hope it gave you some help, some knowledge. Make sure to like this video and subscribe to the channel for future videos and notifications. Thank you.